So here we've got a diagram of all the communicable diseases. It's a bit tricky because different examples need to know different diseases. But I've got a list. I don't know if you guys can see it. I've got some of them here. Not everyone needs to learn all the diseases here. Only some of them are relevant to you. If you want to know which ones, I don't know off the top of my head, what you can do is on my YouTube, I've got video here which has every single communicable disease, and for each exam board it tells you which one you need to know. So Flash Revision Lab on YouTube. Right, I can't see. Yeah, we'll be covering triple and combined content as well. Just give me 1 second. I'm just finding it tricky to get people's comments. So I'll just move this around. So here we got fungi as well. And protist. So there's only one protist you need to know. So exam board, the questions that I've got today are mainly AQA questions. So when it comes to the disease questions about specific diseases, they're only going to be relevant to AQA people. But the other questions on like fighting diseases, those types of questions won't just be for AQA people, they'll be relevant to everyone. Okay, so I'm having problem seeing the comments. I'm going to find it quite hard to reply to some of your comments, guys, but I'll try my hardest. I'm just going to have to keep standing up to see what people are saying, but let's get started. So yeah, like I said, if you want to find out which diseases you need to know, there's a YouTube channel that I've got. Just go to that, watch the video. It's got the specific ones for specific exam boards, and it tells you what causes the disease, how to prevent it, and how it spreads as well for each one that you need to know for each exam board. So it's useful. So just maybe check that out after this. So let's start the 4 different types of pathogens first. Types of pathogen. So we have bacteria. We have virus, and we have protist and fungi. 4 different types. All right. And pathogens are microorganisms that cause disease, specifically communicable diseases. So communicable diseases just means they can be spread. So diseases that can spread. So let's talk about how our body defends against diseases first, before we look at the questions. Let's talk about the external defenses that our body has. So before our immune system kicks in, what do you guys think? What's the outermost defense that we have? Skin. Yeah, thank you. Someone said skin. So skin is the outermost one that we have. So skin basically acts as a barrier. Not only does it do that, it also produces antimicrobial substances. Antimicrobial substances are just ones that kill certain microorganisms like pathogens. After the skin, what other examples do we have, guys? Yes, nose. Let's talk about the nose then. So in the nose, we have two things. We have hairs. What the hairs do is they trap particles that contain pathogens. Also, we produce mucus. Mucus does the same thing, traps pathogens as well. And then I saw someone else say, yeah, this paper one. We've also got it in the, respiratory system and also stomach. So respiratory system is basically our air passages, so our trachea, what we have there is, again, mucus being produced. Does the same thing, traps pathogens. More importantly, we have hairs, but special types of hairs. I saw someone commented, I think. Yeah. It said we also have cilia. So we have cilia hairs which line the air passages. Yeah, I'm finding it tricky to see people's comments, guys. So if you can answer someone else's questions, then please do. This is going to cover triple content and combined content as well. Mostly combined. So what the cilia do, so they're basically special types of hairs. They can move and they trap particles of mucus and they waft it up the air passages, so to the back of your throat. And once it reaches the back of your throat, usually you swallow it. You don't even notice that you're swallowing it. And then it goes to the stomach, and the stomach has hydrochloric acid, which kills the bacteria. So cilia line the air passages and waft particles up to the throat. And then we have the stomach, which produces HCl that kills pathogens. Okay, and then if all of this fails, then that's when the immune system kicks in. That's when your white blood cells kick in. We'll talk about that in a bit. So let's get into the questions. First question, microorganisms cause infection. The human body has many ways of defending it. Describe 2 ways the body prevents the entry. So we've just listed 4-5 ways up there. So you can just pick any 2 of those. So the last thing that we said was acid in the stomach kills pathogens. in food or something like that. What else do we say? We also said the skin acts as a barrier. So you can literally say anything that we listed up there. So you can mention cilia, you can mention the antimicrobial substances. Any of those are fine. Okay, next question. So this question is about specific diseases. So this is AQA. So only AQA people learn gonorrhea. Only AQA people learn measles, I'm pretty sure. Malaria, everyone needs to know about malaria. So do you guys know which ones connect to what? So what is gonorrhea? Gonorrhea is a bacteria. Excellent. Malaria, like I said, this is for everyone. Malaria is not a bacteria. It's a protist. People get confused with malaria. Sometimes they say bacteria, sometimes they say it's a virus. It's a protist. And then finally we have measles. What's measles? Measles is a virus. Well done. Okay, let's get into the proper questions. Malaria is transmitted by mosquitoes. Male mosquitoes can be sterilized so that they're infertile. The spread of malaria is reduced by releasing sterile mosquitoes into the environment. Explain how releasing sterile mosquitoes reduces the spread of malaria. What does the word sterile mean? First of all, before we answer this. This is paper 1 for AQA. I'm not sure about EXO and OCR. Yeah. If they're sterile, it means they can't reproduce. So why is the fact that they can't reproduce mean it's going to reduce the spread of malaria? We have 2 marks, so 2 points. So 1 mark is to literally say it reduces reproduction. Of mosquitoes. And what's the second mark for? Yeah, there'll be fewer. There'll be fewer mosquitoes to pass on the protist. So fewer mosquitoes available to we shouldn't use the word spread. They've already saying spread here, so we'll say to pass on the protist. Okay, good. So this is quite a recent paper. I think it's a few years ago. I forgot what year. All right, so we have 2 different images. One of a fungal spore, rose black spot. Only AQA people need to know that, I think. Fungal spore, so it doesn't matter, it's a rose black spot. It's the fact that it's a fungal spore, and this is a virus. What's the piece of equipment used to see these? Saying microscope on its own is not enough. So yeah, some of you guys have said it. Saying microscope is not enough. You have to say, remember there's 2 types of microscopes. You can have a light microscope and an electron microscope. You have to state which one you're talking about. Remember the difference. The light microscope, this is what you have in your schools. They can see only really big subcellular structures, like the nucleus. They don't have a high magnification. Electron microscopes can see much smaller things. So what you're meant to recognize here is the fact that you can see a virus. Viruses are tiny. They're much smaller than normal human cells. So the fact that you can see this in a microscope tells us it can't be a light microscope. It must be an electron microscope. So that's what you should recognize from this. How many times longer is the fungal spore than the virus? So this is a calculation. So the tricky thing here is we've got different units. We've got micrometers, we've got meters. We want to find out how many times bigger is the fungal spore. So this one's bigger than that. So we need to do some conversions. Let me write down what these conversions are so you guys can note it down. So this is under the topic of microscopes. If you want to go from meters to millimeters, if you want to go from meters to millimeters, what you need to do is you need to times the number by 1,000. If you want to go to millimeters back to meters, you want to divide by 1,000. Then we had millimeters to micrometers. So we will go for millimeters to micrometers. Again, you need to times by 1,000. And then micrometers back to millimeters, you want to divide by 1,000. I hope this is making sense to people. In this question, we have micrometers was one of them, meters was the other. So how would you get from meters to micrometers or micrometers to meters? What's the number you need to times or divide by? It's not going to be 1,000, it's going to be, what do you guys think? 1,000,000, exactly. It's 1,000 times twice. So the easier number to work with, I think, is to go from meters to micrometers. So what you'll do is times by 1,000, times by 1,000 again. So in that question, we'll go to our meters. So this was meters. You want to get this number, times it by 1,000 twice. So 2.5 times 10 to the minus 7 times 1,000 times 1,000, or you can just times it by a million. What does that give us? Can anyone work that out? .25. Thank you very much, Bobby. 0.25. Now we know what this is in micrometers. So this is 0.25 micrometers. So to find out how much smaller it is, we just need to divide the two numbers. So 16 divided by 0.25. And if you do that, it's the same as timesing by 4, you get 64 times 64. So the answer 64. Did that make sense to everyone? Yeah, well done. Those of you who got this, what paper is this? I'm not sure. A few years ago, might be 2020 or 2021. We wish it was 2023. Explain why plants infected with tobacco mosaic virus grow slowly. So this is one of those ones that only some of the examples need to know. So I don't think Edexcel need to know this. OCR, I think they do. I'm not entirely sure. If there's anyone in OCR, let me know if you know whether you need to know this or not. So tobacco mosaic virus, yep, I'm seeing the right answers. So what it does is it discolors the leaves, meaning in a leaves are normally green, right? So if they discolor, it's the chlorophyll that's dying basically. So if you have less chlorophyll, you need to understand what chlorophyll do. Chlorophyll, we use that for photosynthesis. So there's going to be less photosynthesis. Why does that mean? It's going to grow slowly if there's less photosynthesis. So it can't produce glucose. So less glucose, yeah, almost there. Why does less glucose mean they grow less? Because glucose is, first thing you think of when you think of glucose is energy, isn't it? Yeah, exactly. Less amino acids. If there's less glucose, there's less amino acids made, because the plants get the glucose and they make amino acids out of it, and then they make proteins. And proteins is what makes things grow. So that's the last mark. So the first mark is to say there's less chlorophyll in the leaves, which means less photosynthesis occurs. So less glucose made. And less amino acids for growth. This question is higher, but I think it's relevant to foundation as well. How do amino acids help growth? Because amino acids make proteins. If you get loads of amino acids together, it forms a protein, and protein is what helps us grow, helps plants grow as well. If you break down a protein, you get amino acids. So thinking backwards, the plants, they get the glucose they make, they combine it with nitrates to form amino acids. And then if you get loads of amino acids and form a chain, you get protein. Hope that made sense. Okay, next up, let's see what we've got. All right, another question. So we've got a lungworm. It's an infection. It can kill dogs. It's caused by a small worm. And we've got a life cycle of the lungworm here. So it's a disease. Lungworm reproduces in the dog and infects the lungs. The larva is basically young stages of the worm, are coughed up from the lungs and swallowed. Worm larvae pass out in feces and are picked up by snails. And then the dogs touch or eat the snails, and that's how they get the lungworms. Okay, so based on that information, the first question asks, what type of organism is represented by the snail in the life cycle of the lungworm? Which one do you guys think it is? Getting all types of answers here. Right, okay, I'm starting to get the right answers now. Vector. Yes, it is a vector. What is the definition of a vector, anyone? What does a vector do? Let's write that down in our notes. Transport protist, that's one way of saying it, but that's not, there's more to it. Yeah, it's an organism that carries a disease or carries a pathogen without getting the disease itself. So the snail carries the lungworm, but it doesn't get the disease. What's an example of a protist? There's only one I think we learn. What's another example of a vector and a protist that we learn? A protist is just a type of pathogen. Mosquitoes, yeah. With malaria, guys, remember mosquitoes are the one that carry the pathogen around. They don't get malaria. They just carry the protist around, and then when they bite humans, they transfer the protist to them, the pathogen to them, and the humans get the malaria. The mosquitoes themselves don't, so they're known as vectors. So a vector is an organism that spreads and carries a pathogen / disease without getting the disease itself. Okay, hope that made sense. So yeah, that's why it's a vector. All right, suggest how the spread of lungworm disease can be prevented for 3 marks. Let's see what people think. Yep. Killing the snails, yep. So you can kill the snails. That's one thing. Because remember, they're the ones spreading the lungworms. If there's no snails, there's nothing to spread. Same with mosquitoes. That's why in that other question that we did, they were making this mosquitoes fertile so that they can't spread. Vaccine, yes. They can vaccinate the dogs. Well done. One more. If a dog's got the lungworm, what do you need to do to the dog? Instead of vaccinating them, what else should you do to stop it from spreading? Because remember, there's a whole cycle going on here. How can you prevent other dogs from getting it? Quarantine, exactly. Isolation, yes, good guys. Isolation. So you can isolate the infected dog. Good. Okay, next. We've got another question about malaria. I think it's describe 2 ways to control the spread of malaria. So it's linked to this one. What are 2 ways we can control spread? Mosquito nets, yes, good. And mosquito nets. Insect repellent or pesticide, good. Or you can stop mosquitoes from breeding as well. I know it's not asking for 3. But you could say that as well. Next one. All right. So next question. we have number of people with measles. It is a question about measles, but this could be any disease. You could get a question about any disease similar to this. Measles, only AQA people learn, but imagine you get a graph like this and there's a vaccine. It doesn't matter what the name of the vaccine. All of this happens. So compare how effective it is introducing the measles vaccine and introducing the MMR vaccine. So they're two different vaccines. All right, so you don't need to know what each one does. AQA people do need to know what MMR is, but there's two different vaccines. That's the main point there. We've got two different vaccines. So you need to compare the graph after the first one's introduced and then after the second one's introduced. It's asking to compare how effective the first one was compared to the second one. What are some of the points we could say? So compare means you don't need to think of differences, not just differences. You can also state similarities. I'm going to stand up for this to see what you guys are saying. Compare range. Less people with measles. Both reduce it. Yep, that's what I was looking for. So look at the general trend. I know it's going up and down, up and down. That's going to confuse a lot of people that it's going up and down. When you get questions like this, try and think of the general trend. The general trend is basically doing this, isn't it? It's going across, something like that. So don't worry about the small increases and decreases. So both cause it to decrease. That's one similarity. Let's think of some differences. What's the difference between the first one and the second one? Yes, MMR, the second one, causes it to decrease to 0. Well done. See that? That completely, let me rub this off. That completely eradicates measles, whereas the first one didn't. Eradicated measles to 0. Another thing, the measles caused a bigger drop. Look at the difference here. Measles, if you read the graph, it starts around here, doesn't it? And it drops it to around here. Look at how many thousands of people that is. That's about 400,000 people. MMR, when MMR is introduced, it's around here, isn't it? And that reduces it to 0. So there's not a big decrease, but the measles vaccine, there was a big decrease. So it did reduce it to 0, but this one had a much bigger decrease. It had a bigger effect. So the measles vaccine caused a bigger decrease. Well done. Right. This is a question just for AQA people. Other than measles, which two diseases does MMR vaccine protect against? What does MMR stand for? Can I go back? Yep, let me go back for 1 second while people answer that. What does MMR stand for? Yep. Well done. So the first M is for measles, second one's mumps, and last one is rubella. So we've already talked about measles, so this got mumps and rubella. Right, next we've got immunizing someone. So we immunize someone against measles, a small quantity of inactive measles pathogens, so this is relevant to everyone, is injected into the body. What happens to the body after immunization? So when they say immunization, they mean vaccines. So what does a vaccine do? Let's add this to our notes. This vaccinations, so you inject a dead / inactive pathogen, and the inactive pathogen has an antigen. Yeah. An antigen, guys, is basically a marker that pathogens have. It's like on the outer part of a pathogen, and every pathogen has a different antigen. And that's how our body recognizes that it's a different type of pathogen, it's a new type of pathogen. Yeah, so it contains an antigen, and then what does our body do? Our body basically treats it, even though it's a dead pathogen, it treats it as if it's a real pathogen that's about to cause us harm. So our body thinks it's attacking our body. So it tries to fight it off, and the way it fights it off is by producing antibodies. And those antibodies, they basically lock onto the antigen and they destroy the pathogen. Right? These ones are already dead, but the most important thing is once they have found how to fight this pathogen, so even though it's a dead pathogen, once it's figured out how to fight the pathogen, if this pathogen was to ever enter the body again, our body would have the right antibodies to fight it. So if this pathogen was to enter again, it would be able to rapidly produce the antibodies to kill the pathogen. Right? So some points we could say, so you inject inactive pathogen, which contains an antigen. The white blood cells or lymphocytes produce antibodies to kill the pathogen. Well, it's already dead, isn't it? So you're not killing it, it's just producing antibodies. But if the pathogen was to re-enter, the important thing is the body would produce antibodies, but rapidly and with a greater amount. The white blood cells, these are called memory cells, the ones that live on. So white blood cells produce antibodies rapidly and in a greater amount. Okay. Yeah, so more accurately, what happens is the antibodies lock onto the antigens, and then other white blood cells called phagocytes come and engulf them, they eat them. Yes. Okay, so this question here, to answer that, for 3 marks, first thing, you get a mark for mentioning white blood cells or lymphocytes. So white blood cells. So you can either say white blood cells, you can say lymphocytes or memory cells produce antibodies if, we'll say rapidly and with a greater quantity, so greater amount, if the pathogen was to re-enter. Hope that makes sense, guys. Let me see. You guys got any questions? Okay. In this question, you don't need to talk about what the antigens do. It's only 3 marks. Could you put that? It puts antitoxins. No, not antitoxins isn't relevant for measles because measles is a virus. Toxins are only produced by bacteria. Higher or foundation? This is a higher question. I'm not sure if it's relevant to foundation. What do antigens do? Antigens don't really do anything. They're just, imagine you got a pathogen. They're just markers that are on the outside of the pathogen. And every pathogen has a different marker, has a different type of antigen. So these triangular things are the antigens for one type of pathogen, and on another one, it would be like a different shape. Obviously, it's much more complicated than this. So that means our body recognizes these 2 pathogens as completely different. So when they produce antibodies, the antibodies need to basically connect or latch onto these antigens. So let's say we had 2 different antibodies, one that looked like this. Think of like enzymes. It's specific. These are specific, like enzymes. So we have one like this, and then we have one like this. This antibody is going to be for this pathogen because it latches on to the correct shape, like that. This one would latch onto the square shape, like this. So if you've got a new pathogen coming into your body, you need a new antibody to fight it. And what they do is they inject a pathogen which has these antigens on the outside, but our body thinks it's a real pathogen. So it tries to make these antibodies fight it. And once they're all gone, we keep the antibodies, or we keep the white blood cells which produce that antibody. So if this was to ever enter again, these would come, and they'll come in greater quantity. So you won't even get ill because they'll wipe out the disease, the pathogen, before you get the disease. I hope that makes sense. So these questions are AQA paper 1. Next question. People may be immunized against diseases using vaccines. Which part of the vaccines stimulate the body's defense system? Exactly what we were talking about now. So it's just a dead or inactive pathogen. This one's quite an old question. I don't think you'd get 2 marks for something like this anymore. Dead pathogen. You get 1 mark for dead, 1 mark for pathogen. A person has been vaccinated against measles. The person comes in contact with the measles, does not catch measles. Same is the same 3 points as before. So white blood cells produce antibodies rapidly, and it destroys the pathogen if it was to re-enter. Yeah, I'm not going to write that down again. It's a repeat question, basically, from another year. Let's go on to the next one. Okay, so here, carrying on. A man catches the disease. The man has not been immunized against the disease. Doctor gives him antibiotics. What do antibiotics do, guys? Got tiny space here. Let's write that down here. Yeah, kill bacteria. Only bacterial diseases. Yeah, so if you have a viral disease, antibiotics are going to be useless. So the question asks, wasn't this one? Oh, it hasn't asked anything yet. So this is when the man takes the antibiotics. These are the number of bacteria in the body. So you can see as soon as he takes the antibiotics, it's still rising. So it must have been rising for a bit. Starts taking the antibiotics, and then it rises, and then it drops as he takes it, because it's starting to kill the bacteria. You can't use antibiotics for fungal diseases. No, you can't. You need fungicide to do that, something different. 4 days after starting the course of antibiotics, the man feels better again. It's important the man does not stop taking the antibiotics. Explain why. Use information from the graph. What do you guys think? I will post this, guys. I'll post this on YouTube. Don't worry. Yeah, Flash Revision Lab. I've posted last week's lives. So after this, at some point this week, I'll be posting this one with the sheet, so you can go and do the sheet as you do the video. Not everything was killed. Yeah, so let's use the graph. It says after 4 days, he feels better. Okay, the reason he feels better is because there's not that many bacteria left in his body. So most of them have died, haven't they? But they haven't all died. And imagine the antibiotics have killed probably the weaker ones first. It hasn't killed the stronger ones. So the only ones surviving right now, these ones, are the stronger ones. So the man should carry on taking the antibiotics so that this goes to 0. Because if he's left with just the stronger ones, even though he's got a low number, they're going to reproduce again, and they're going to be even more difficult to kill. So it's going to get worse. So that's why it's so important people finish the antibiotics when they take it. So for 2 marks, we can say from the graph, it says use information from the graph, so we can say there are still bacteria in the body which can reproduce. Yeah. Yeah. It's going to finish soon, guys. I'm just going to do a few more questions. Occasionally, a new resistant strain of pathogen appears. The new strain may spread rapidly. Explain why. What do you guys think? No antibiotics. Yes. It's a new strain. So there's not going to be any antibiotics that really work on it. It's not the body that produces antibiotics. The body produces antibodies. Antibiotics are drugs that we take to kill bacteria. Yeah, so the antibiotics would not kill the new strain. And what will happen is it means the bacteria will reproduce. And then what you're going to get is you're going to get a population of resistant pathogens. So all of these which are resistant, they're just going to increase, and antibiotics are going to be completely ineffective against them. So population of the resistant pathogens increase. When will I go live again? I'll go on tomorrow at 8 p.m. for chemistry. We're going through energy and reactions. So exothermic, endothermic. And then Sunday we'll do physics. All right, next one. What type of pathogen causes malaria? We've done this already. Protist. 2 methods to prevent people catching malaria. So we gave 2 methods, but we didn't give a reason for why each method works. So let's try and talk about some of these then. So we said nets. So what would be the reason for using mosquito nets? So yeah, nets for mosquitoes. What's the reason for them? To stop the mosquitoes biting, exactly. To stop mosquitoes biting. And another one. Yeah, you can say insect repellents. Yes. And the reason for insect repellent is because they're less likely to bite you. Again, same thing. So less likely to be bitten. You guys said some other correct ones. So you could also say malaria. Yeah, malaria tablets, good, because that's going to kill the pathogen, it's going to kill the protist. You can say vaccine, yeah, so that means more people are immune to it, so less people will spread it, or there's less people for the mosquito to spread it with. What's the difference between a protist and a vector? A protist is the actual pathogen. It's what's causing the disease. The vector is the mosquito. The mosquito is only spreading the disease. It's not causing the disease. So this mosquito won't get malaria. All it does is it carries the protist, the pathogen, from one human to another. So when it bites one human and then it goes to another human, it's spreading the protist. That's the difference. Hope that made sense. All right, we'll make this the last one. Describe 2 differences between bacterial and eukaryotic cells. So this is more of a cells question for 2 marks. does not have a nucleus. Yep, bacteria. Do not have nucleus. And bacteria are smaller. Yep. And have plasmids. Perfect. Well done, guys. Right. Let me show you our notes so you can screenshot. That's everything we did. So this, I know you can't read it properly. Like I said, it's on YouTube. We zoom into all of these and talk about how it's spread, what type of pathogen causes it, and how to prevent it. So take a look at that if you don't memorize or you don't know all the diseases that you need to know for your specific exam board.